Hey everyone! If you are getting ready for the CUET exam, then you are in exactly the right spot. Today, we're going to tackle a topic that is an absolute goldmine for marks, the Hardy-Weinberg principle. Let's dive right in and make sure you nail every single point you need from this. Okay, so let's start with a bit of a puzzle. We're always told that evolution is this constant, ongoing process, right? Things are always changing. So wait a sec. Why don't we see the genetic makeup of every population totally shift with every single generation? It's a great question, and the answer is the absolute key to understanding everything we're about to cover. And why should you care about this? Really? Because, and I'm not kidding, the people who write this CUET exam love this concept. Seriously, mastering Hardy-Weinberg isn't just about understanding some cool biology, it's about locking in guaranteed marks. Stick with me, and I promise you're gonna get them. So, what's actually going to be on the test? This right here, this is our game plan. You can bet you'll see at least one math problem using the Hardy-Weinberg equation. Then they're going to hit you with the concept questions about the conditions for equilibrium, the founder effect, the big difference between genetic drift and gene flow, and of course, the roles of mutation and recombination. This is our checklist for success today. Okay, so let's get right to the core idea. The best way to think about the Hardy-Weinberg principle is as an evolutionary standstill. Imagine you could hit a giant pause button on evolution. This principle describes that baseline, that perfect theoretical situation where absolutely no change is happening. And here's the official definition for you. It comes from two brilliant scientists, G.H. Hardy and Wilhelm Weinberg, who actually figured this out completely separately. All it says is this. In a really large, ideal population, the allele frequencies, the different versions of a gene, will stay exactly the same, generation after generation. The most important part to remember, this is the definition of when evolution is not occurring. All right, let's get to the part that sometimes makes people nervous, but trust me, it's super simple when you break it down. The math. And this isn't just some random formula. This is literally the equation that describes genetic stability. First up, we have the easy part. For any gene that has two alleles, a dominant one and a recessive one, their frequencies have to add up to 100%. Or as we say in math, they have to equal 1. So we call the frequency of the dominant allele P and the frequency of the recessive allele Q. So P plus Q equals 1. That's it. Simple as that. Now let's bring in the main event. This equation here describes the actual individuals living in the population based on their combination of alleles. It probably looks familiar from math class, right? And just like the last one, it all adds up to 1 because it represents every single individual in that population. And this table breaks it down perfectly. That p-squared term, that represents the frequency of the homozygous dominant individuals, the ones with two copies of the big A allele. The q-squared term, that's the frequency of the homozygous recessives, the little a guys. And that 2pq in the middle, that's our heterozygotes, the ones carrying one of each allele. You've got to have this table memorized, it's non-negotiable. So how do you actually use this on the exam? Here is your step-by-step playbook. Number 1, find the piece of information they give you. Almost always, they'll tell you the frequency of the recessive individuals, which is your q-squared value. Your very first move is step 2. Take the square root of that number to find q. Then it's easy. Use p plus q equals 1 to find p. And once you have p and q, you can find anything they ask for, like the frequency of heterozygotes, which is 2pq. I need to say that one more time because it is so important. This is a massive CUET tip. The single biggest mistake students make on these problems is forgetting to take the square root of q squared. They just use the number from the problem as q, and their whole calculation is wrong from the start. Don't be that student. Always find q first. Okay, so we've just defined this perfect imaginary world where there's no evolution. But the real world is messy, right? So let's talk about the things that actually mess up this perfect balance. The forces that push populations out of equilibrium and make evolution happen. Let's meet the 5 big disruptors. And here they are. Think of these as the 5 engines of evolutionary change. If any one of these is happening, gene migration, genetic drift, mutation, recombination, or natural selection, then that perfect Hardy-Weinberg balance is broken. The allele frequencies will start to change, and boom, that's evolution happening right there. Now, let's zoom in on 2 of those factors that students always get mixed up, genetic drift and gene flow. They sound so similar, but they are totally different things. Let's clear this up for good. Okay, look closely.
Genetic drift is all about random chance. Pure luck. It has the biggest effect in small populations, where a random event can totally change the gene pool. On the other hand, gene flow is just another name for migration. It's the literal flow of genes when individuals move into or out of a population. This introduces new alleles and can make two separate populations more similar to each other over time. And a classic example of genetic drift that the CUET loves to ask about is the founder effect. Just imagine a few individuals, our founders, get separated from the main group and start a new population somewhere else. Just by pure chance, that small group of founders might have a very different set of allele frequencies than the original population they came from. So what about those other factors? Well, think of mutation as the ultimate source of newness. It literally creates brand new alleles that never existed before. Recombination doesn't create anything new, but it shuffles the existing alleles around into new combinations, kind of like shuffling a deck of cards. Together, these two provide all the raw genetic variation, the material that natural selection has to work with.
All right, you've got the core concepts down. Now it's time for the CUET Power-Up section. This is where we lock this info into your brain for exam day, using some simple memory tricks and pointing out the most common mistakes people make. Let's lock these in. For genetic drift, just think of a tiny boat drifting by chance at sea. It has a big effect on a small boat. For the founder effect, remember that a few founders start a new colony. And gene flow is easy. Genes are flowing from one population to another through migration. Got it? These little word associations are going to be a lifesaver on the exam. Okay, listen up. This is super important. We need to avoid these common mistakes. Number 1, we've hammered this home, forgetting to take the square root of q squared. Number 2, confusing flow with drift. Remember, flow is migration, drift is chance. Number 3, thinking drift is a big deal in large populations. Nope, its power is in the small ones. And finally, don't forget the simplest piece of the puzzle, p plus q equals 1.
Okay, we're in the final stretch. Time to lock in all this knowledge with a quick challenge. I'm about to hit you with a few QET-style questions. Get ready to test yourself. All right, let's see what you've got. Question 1. Which of these is most likely to mess with genetic equilibrium? A, B, or C? Pause and think about it. The answer is C, mutation. The other two actually help maintain equilibrium. Okay, question 2, a little math. The recessive phenotype is 0.09. What is the frequency of the recessive allele Q? Pause now and solve it. Did you get it? The answer is A, 0.3, because you have to take the square root. Last one. Genetic drift has the strongest effect in A, B, or C. The answer is, of course, B, small populations. So, how'd you do? Time for a rapid-fire revision. These are the absolute must-know facts. Ready? Hardy-Weinberg means genetic equilibrium, which means no evolution. If a population deviates from Hardy-Weinberg, it is evolving. The key equation is p squared plus 2pq plus q squared equals 1. Genetic drift hits small populations the hardest, and mutation is the ultimate source of all new alleles. Got it? So, I'm going to leave you with one final question to really tie this all together. Why does a deviation from the Hardy-Weinberg numbers prove that evolution is actually happening? Think about the fundamental definition of evolution. I'd love to see your answers in the comments below. And hey, if you're serious about crushing the CUET and you're ready to master the next big topic, make sure you're subscribed. We've got more game-changing explainers on the way. Keep up the great work.